Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled The Balance of Safety and Efficacy, Understanding Target Antigen Density in Cancer Treatments with the IndEx-2 Inducible Cell Line Platform. My name is Lawrence Howes and I'll be moderating today's presentation. I'm delighted to be joined by our guest speaker, Dr. Timothy London, Scientific Director of Molecular Biology at Antibody Analytics. This webinar will discuss the IndEx-2, an advanced in vitro cell-based platform engineered to modulate the expression of one or two target antigens across a range of biologically relevant levels. Following the presentation, we will have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these in the box to the right of your screen. Without further delay, I'd like to hand over to our speaker, Timothy, and I'd like to thank them again for presenting for us today.
Thank you everyone for joining us today for this webinar, where I will describe our recently developed IndEx-2 system and its use in assessing the impact of antigen density on the efficacy and safety of immune-directed therapies. Before I begin, I'd like to thank Select Science for hosting us today, and also Agilent for facilitating and sponsoring this webinar. Before I get into the scientific portion of my talk, I'd first like to introduce Antibody Analytics and what we do. We are an immunology-focused CRO based in Scotland in the UK. We are located approximately 20 miles outside of Glasgow in a town called Motherwell, where our labs are located. We have more than 90 employees and we're still growing. In terms of our expertise, we're a world leader in human immunology research service provision for the assessment of immuno-oncology, autoimmunity, and biosimilar therapeutics. We apply a unique combination of immunology, bioanalytical, and molecular biology expertise to establish advanced experimental solutions to assess the safety and efficacy of therapeutics. We specialize in antibody-based drugs and advanced medicines, including immune cell therapies, and we support a global customer base who operate from the earliest phases of drug discovery to the clinic. One of the key challenges in developing tumour-targeting therapeutics lies in identifying optimal tumour-associated antigens. Often these targeted antigens are not cancer-specific. They are overexpressed on cancerous tissue, but can also be found in healthy tissues. This heterogeneous tumour antigen expression not only makes efficient treatment more difficult, but also contributes to immune evasion by tumours that downregulate the expression of the target antigen, so-called antigen escape. This challenge applies to multiple immuno-oncology therapeutic modalities, including immune cell therapies, immune cell engagers, ADCs, and conventional monoclonal antibodies. The expression of target antigens in normal tissues can lead to off-tumor toxicity, which is a common side effect associated with many immune-engaging therapeutics. In particular, on-target off-tumor toxicity has become a significant concern, and regulatory guidelines have been established to assess the undesired targeting of healthy tissues that express the target antigen. And you can see that here in a snapshot from the current FDA guidelines for the development of CAR T-cell therapeutics. The conventional approach to assess the impact of antigen density on the efficacy of therapeutics is to use a panel of cancer cell lines that express differential levels of the antigen of interest. These cells are employed in functional assays, and it is typically expected that the observed response should be proportional to the number of receptors. However, this approach has various limitations. Firstly, the cost of setting up an appropriate panel of cancer cell lines can be prohibitive, and can reach tens of thousands of dollars if licensing costs are included. Secondly, the range of expression in even a large cell line panel can be limited, which does not allow determination of the threshold of activation of the therapeutic. And lastly, the cell line background can introduce variability, as intrinsic differences between cell lines can render them susceptible or resistant to a given therapeutic in a manner that's independent of the antigen expression level. I'd like to draw your attention to this publication that effectively highlights the limitations of conventional approaches when assessing antigen density in the context of CAR T therapies. In this study, the authors aim to fine-tune the effector function induced by HER2 and EGFR targeted CAR T cells by modulating the affinity of the antigen binding moiety of the receptors. To evaluate the different HER2 CAR-T constructs, they employed a panel of 11 cancer cell lines. However, as you can see, despite having 11 different cell lines, only 3 levels of HER2 expression can be observed: high, medium, and low, with minimal differences within the groups. The authors generated 4 HER2 targeting CAR-T constructs based on the trastuzumab antibody, modifying them to create constructs with reduced affinity to HER2 compared to the original antibody. When testing the ability of the generated CAR-T cells to produce interferon gamma as a measure of activation against the cell line panel, the results were quite surprising. Focusing on the CAR-T based on the 4D5-7 antibody, there is no clear correlation between antigen density of the target cell line and the biological response for this CAR-T. Interestingly, a much higher interferon gamma response is induced against the PC3 cell line, which actually expresses low levels of HER2 compared to SCOF3 or SKBR3 cells that express high levels of the receptor. However, when the authors assessed CAR-T cells against a common genetic background, they observed a clear dependency of antigen expression on biological activity. They achieved this by transfecting different amounts of HER2 mRNA into K562 cells, resulting in HER2 expression proportional to the amount of mRNA transfected. In this case, a clear dependency of the biological effect induced after CAR-T engagement on the levels of target antigen can be observed. This study underlines the importance of considering cell line background when assessing antigen density and its impact on the safety and efficacy of therapeutics. In order to address the limitations of the conventional approach to assessing antigen density, a better system would have the following characteristics: a single cell background to ensure the absence of confounding factors from heterogeneous cell populations, a large dynamic range of expression. This would enable the assessment of antigen abundance across a wide range of expression levels from hundreds to hundreds of thousands of receptors per cell, providing a more comprehensive analysis. Finally, titratable expression. The ability to control antigen expression levels is crucial for understanding the impact of the varying densities on therapeutic efficacy. And finally, the availability of a negative control. A negative control is essential for establishing a baseline and validating experimental outcomes. We aim to incorporate all of the previously mentioned characteristics into our solution, which is the IndEx-2 assay platform system. This system has 3 main components. At its core is an inducible antigen expression cell line system, which I'll come to in a second. That is coupled with our suite of functional assays that we have in-house at Antibody Analytics, which includes systems such as the Excelligence real-time cell analyzer system, flow cytometry capabilities, and the ability to analyze cytokine production. And then finally, we have a set of analytical techniques which allow us to calculate a threshold of activation for a given therapeutic. For those of you who aren't familiar with the Excelligence real-time cell analyzer system, I'll just give you a quick overview of it in the next slide. The Agilent Excelligence RTCA system is an impedance-based measurement system which measures the attachment of adherent target cells via electrodes running through the base of the wells where the cells are grown. Upon addition of effector cells or a test drug, cytolysis can be detected by the detachment of the target cells from the base of the wells, which causes a reduction in the impedance value measured through the electrodes. This technique can be combined with other readouts such as flow cytometry and cytokine analysis, and we employ it for multiple therapeutic modalities, including monoclonal antibodies, immune cell engagers, CAR-Ts, and ADCs. The system that we employed to control the expression of target antigens in our IndEx-2 system is based on something called chemically induced proximity. This is actually a system that is derived from plants, in particular Arabidopsis thaliana, where it functions as a stress response pathway. At its core, it is a protein heterodimer that only heterodimerizes in the presence of a small molecule. by using this protein heterodimer pair and attaching a DNA binding domain to one of the halves and a transactivator to the other, and placing the DNA recognition motif for the DNA binding domain upstream of your target antigen of interest, you can create a system in which, in the presence of the small molecule shown here, the transactivator is brought into proximity of your target antigen of interest, which then switches on transcription and translation. I say switch, this happens in a very dose-dependent manner. So the more you add of the small molecule, the more expression you get of your target antigen of interest. Most of the data that I'm going to show you today is from a system that responds to abscisic acid. The reason we chose this one is that we got high titratability, and also abscisic acid is completely inert to mammalian cells. To build the system, we use the CHOK1 cell line as our background. The reason we chose this is that it's a non-human cell line, so obviously it doesn't express human antigens, therefore it functions effectively as a blank canvas. To build the system, we introduced via lentivirus what we call the induction cassette, which carries the protein heterodimer, which I described in the previous slide, and then a second construct which carries the target antigen of interest with the DNA binding domain recognition motif upstream. To exemplify the system, we chose a number of target antigens which we believe are industry relevant, so EpCAM, HER2, BCMA, and CD19. And you can see here in these graphs that we're able to control the expression of these target antigens over a wide expression range. The pattern of expression is different for each of them, but we believe that this is most likely due to both differences in size of the receptors and also receptor recycling kinetics. On the right-hand side, we have some data from expressing two antigens in the same cell line background. So we have CD22 under the control of the abscisic acid system, and then CD19 under the control of the indole-3 acetic acid system. We've been able to show that these two systems are orthogonal, but this is still a work in progress as we are hoping to optimize both the background and levels of expression that we get from these two systems in the same cell line. So how can we use the IndEx-2 system to assess therapeutics? The first example I'd like to give you is the use of the system in comparing high and low affinity HER2 targeting CAR-Ts. So in this experiment, we had 4 different HER2 targeting CAR-T constructs, 2 high affinity CAR-T designs, one with CD28 as the co-stimulatory domain and one with 4-1BB. And then we also had 2 low affinity CAR-Ts, again with a CD28 and a 4-1BB co-stimulatory domain. We use these constructs in the following assay setup. Our target cell line was the CHOK1 inducible HER2 cell line, induced over a range of expression levels of HER2. And then we had T cells isolated from 3 donors into which we transduced the CAR constructs that I just showed you. The assay format was using the Acceligent system, so the readout was target cell cytolysis. When we plot the level of cytolysis against the HER2 receptors per cell for the low affinity and the high affinity CAR-T constructs, you can see that the higher affinity has a much sharper increase in cytolysis at the lower levels of HER2 expression compared to the low affinity. And this is what we would expect to see. What is also interesting is that the CD28 and 4-1BB co-stimulatory domains do not actually make that much difference in terms of the level of cytolysis achieved by either of the 2 CAR-T constructs. The next example I would like to focus on is the targeting of CD19. As you are no doubt aware, there are several approved CD19 targeting therapeutics. Even though these therapeutics are very successful, a proportion of patients that are treated with them do not respond or relapse after a short period of time. One of the reasons that this happens has been linked to the levels of CD19 expression by these patients. In this publication shown here on the left, Spiegel and colleagues tried to correlate the levels of CD19 expression to the response of Yescarta treatment in large B-cell lymphoma. By employing quantitative flow cytometry, they defined that there is generally a higher chance of resistance when the levels of CD19 expression falls below approximately 3,000 receptors per cells. Beyond CAR-T, similar reasons for resistance has been reported for Blincyto for B-cell acute lymphoblastic leukemia. So using these publications as a guide, we can define a broad area of expression levels that are linked to lower or higher risk of resistance to therapy. Therefore, an ideal CD19 targeting therapeutic would retain efficacy at all levels of CD19 expression. The ability to target a wide range of CD19 expression in leukemias, including low levels of CD19 expression, was one of the factors that led our collaborators, Cullinan Oncology, to develop the CD19 targeting T-cell engager known as CLN-978. This has a very high affinity binding moiety for CD19 and includes an HSA binding domain for extended serum half-life. It is predicted to work in very low expression levels of CD19, and as you can see here, it is effective at low concentrations in terms of cytolysis of target cells, interferon gamma, TNF-alpha production, and T-cell activation as measured by CD8 and CD25 positivity. This therapeutic has the potential for use in relapsed B-cell malignancies where there has been CD19 antigen loss. So given that CLN-978 would be expected to have a low threshold of activation in terms of CD19 expression, can we use our IndEx-2 system to assess this? So the experiment we set up used the CHOK1 inducible CD19 cell line as the target cell line, and then we isolated T cells from a panel of 10 healthy donors and compared CLN-978 to another CD19 T-cell engager, blinatumomab or Blincyto, and the readouts we used were cytokine production as measured using the Luminex system, and target cell cytolysis as measured using the Agilent Excelligence RTCA system. For this panel of 10 healthy donors, CLN-978 causes a consistently higher level of cytolysis across the entire range of CD19 expression levels expressed from our target cell line. The biggest differences are seen at the lower levels of CD19 expression, with the level of cytolysis becoming more comparable at the higher levels of CD19. A similar scenario happens with the interferon gamma production by the effector cells, with CLN-978 causing a consistently higher level of interferon gamma production across the entire range of CD19 expression from our target cell line. In order to determine the receptor threshold of activation for the two molecules, we used an alternate analysis of the 10 donor data panel, employing something called a receiver operating characteristic, or ROC analysis. This is a technique originally developed during the Second World War for interpreting radar signals. However, ROC analysis has since found widespread applications in various fields, including diagnostic test development, where it is used to distinguish between positive and negative effects. However, it can also be used to define a threshold above which something is considered true. In this case, we used it to define the threshold of receptor activation, employing cytotoxicity or interferon gamma production as the readouts, and then compared CLN-978, shown in grey, against Blincyto, shown in the red dotted line. We then used a statistical technique called Uden's J index, or Uden's J threshold, to define a threshold of activation. This corresponds to the point on the ROC curve that maximizes the difference between the true positive and the false positive rates. So for CLN-978, both readouts, cytotoxicity and interferon gamma, correspond to a threshold of activation of approximately 80 receptors per cell. However, for Blincyto, the threshold of activations are quite different between the two readouts. For cytotoxicity, the threshold of activation is around 850 receptors per cell, and for interferon gamma, the threshold of activation is closer to 8,500 receptors per cell. However, this analysis is useful in that it clearly shows that the Cullinan molecule has a much lower threshold of activation compared to a conventional therapeutic such as Blincyto. Given the data I've just shown you, we can then speculate that the Cullinan molecule, CLN-978, can be potentially used in cancer scenarios where the CD19 expression level would normally be associated with a high probability of resistance. This is in contrast to blinatumomab, where its range of efficacy in terms of the levels of CD19 expression are normally correlated with a lower probability of resistance. I should probably point out here that this scenario is fairly unique to CD19. If the target was something else such as HER2, you would then be looking for your therapeutic to have its threshold of activation above the level of receptors per cell that you would normally see on healthy tissues. We have also used our IndEx-2 system to assess other therapeutic modalities. So shown here on the left is a bispecific CAR-T targeting CD19 and CD22, and we use this with our target cell line, the CHOK1 cell line that I showed you towards the beginning of the talk, which expresses CD22 under the control of the abscisic acid or ABA system and CD19 under the control of the indole 3 acetic acid system. And you can see when we induce only CD19, shown in the blue here, we get this response from the CAR-T, which is expressed on our JERCAT NFAT luciferase reporter cell line, so the readout is luminescent as a measure of activation of the CAR. When we induce CD22, we get a higher level of activation, but when we induce both CD19 and CD22 at the same time, we see this additional effect shown here in purple. This is interesting because the CAR in question is actually an OR CAR rather than an AND CAR. That is, it requires the presence of CD19 or CD22. It doesn't require both, but even so, in the presence of both of its two targets, we are able to see this additional effect. On the right is an example of an antibody-drug conjugate. In this experiment, we compared two HER2 targeting ADCs, both based on the trastuzumab antibody, but carrying different payloads. And you can see here when we used our inducible HER2 expressing CHOK1 cell line as the target, at the lower levels of HER2 expression, the level of cytotoxicity caused by these ADCs is very similar. But at the higher levels, or the highest levels of HER2 expression, you can see quite clearly that the ADC carrying the mptansine payload has a significantly higher level of cytolysis. Again, we would argue that our system is useful here because if you had a cell line that expressed a level of HER2 in the lower levels, you would not be able to see the difference in cytolysis caused by these two ADCs. That only becomes apparent when you look at the cytolysis across the full range of expression of HER2 receptors per cell. So in summary, the IndEx-2 system is an in vitro system that allows the estimation of antigen thresholds for the initiation of effector functions. I hope I have convinced you that it is useful for assessing on-target off-tumor side effects, and that it is also applicable for multiple types of therapeutics, including monoclonal antibodies, T-cell engagers, ADCs, and CAR-Ts. I'd just like to finish up by acknowledging our collaborators, Cullinan Oncology. This work was funded by a grant from Innovate UK, and all of the work was performed within the Antibody Analytics Research and Development Department by Hannah, Amy, and Kieran. And with that, I will be happy to take any questions.
Thank you for that very informative presentation. It is now time for our question and answer session. So if you do have any further questions, please do send those in. You can submit these by typing into the questions box to the right of your screen. So we've had a couple of questions come in. To begin with, what is the range of receptor expression that can be achieved with this approach?
So it depends on the receptor in question, but we tend to see that we can get from around a few hundred receptors all the way up to around 1 million receptors, or in some cases slightly more. For example, HER2 seems to express very well with our system. We get slightly over 1 million receptors per cell.
Excellent, thank you very much. The next question we've had in here is, what cell line backgrounds can be used for generating IndEx-2 cell lines?
So we've mainly used the CHOK1 cell line to develop the system, as it's a non-human cell line. So obviously it doesn't express human antigens and effectively is a blank slate. However, the system should work in any cell line that's amenable to lentiviral transduction, which is our mode of delivery. The only consideration really with using a human cancer cell line, for example, is whether or not it already expresses the target antigen of interest. But that is not, that wouldn't completely stop you using that cell line, because it is possible to make a knockout of the target antigen of interest and then introduce it back into the cell line again under control of the IndEx-2 system. So then you have your target antigen of interest in a relevant cell line background, now under control of the IndEx-2 system.
Great, thank you for clarifying that one for us. We have had another question come in here. Does the small molecule inducer affect the growth or viability of either the target or effector cells?
The short answer is no. It's actually pretty biologically inert in mammalian cell culture. We have looked at the effect on a number of cell lines, so CHOK1 and HEC293, for example, and it has up to very high concentrations, higher than we use to actually induce expression from our system. We haven't seen any impact on the viability or growth of cells. We've also looked at the effector cell side. So we expanded T cells from PBMCs in the presence of the small molecule inducer, again at high concentrations, and we didn't see any impact on the expansion of T cells or indeed the characteristics as measured by antigen expression such as CD4 and CD8, etc.
Great, thank you very much. So we are going to wrap it up there. I'd like to thank our expert speaker, Timothy, for the informative presentation and discussion, and a big thank you to everyone for joining us online. We hope you found this a worthwhile session. If you do have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speaker. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it'll be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.